Welcome everybody to this online presentation. My name is Jelger Herder and I work for RAVON, Reptile Amphibian Fish Conservation. And we did this research together with SpyChain, who did the eDNA analysis. And our talk will be about eDNA-metabarcoding fish in relation to the Water Framework Directive indices. To determine the ecological health and quality of water bodies, the Water Framework Directive recognizes fish as a biological quality element. And therefore, fish is monitored. In the Netherlands, this is done by electrofishing, pike nets, sea nets, and on the large rivers and lakes, we also use trawling. And all those methods are written down in strict protocols in the Handbook of Hydrobiology from the STOA. So some years ago, we started with the eDNA-metabarcoding fish studies in Europe. This study is published in Valentini et al. And in this study, we tested the method in rivers, streams, ponds, lakes, canals, and also in marine environments, in France, in the Netherlands, and in Denmark. And this study showed the high potential of eDNA-metabarcoding. It showed basically that we could detect the same amount or even more species as caught with traditional methods. So in parallel, we set up a large project in the Netherlands to look if we could use the eDNA-metabarcoding for the Water Framework Directive. And first, we focused on transect level. So for the Water Framework Directive, for each water body, several transects are fished, maybe 5 up to 40. And we selected 55 of those 250-meter transects all over the Netherlands in different water bodies. And we compared them to the traditional monitoring that was done by sea net fishing and electrofishing. So in this graph, you see the results for the number of species caught by both methods. So in yellow, it gives the number of species detected with eDNA, and in blue, it gives the number of species directed with the Water Framework Directive monitoring with sea nets and electrofishing. So on average, we found 1.6 times more species were detected with eDNA compared to traditional monitoring. And more important, when we looked at individual species, all species had equal or higher detection rates with eDNA. So those results were very good. And when you see in the graph on the right, in flowing water, the difference was highest. This is what you can imagine, because in flowing water, the eDNA can travel a little bit from upstream. So you're actually sampling a larger transect. But also in stagnant waters like lakes, it's still 30% higher detections with eDNA. So then in 2016, we upgraded this project because for the Water Framework Directive, they don't look at transects, but they look at complete water bodies. So we sampled 9 complete water bodies in the Netherlands, both with eDNA and in parallel to the Water Framework Directive monitoring, mostly with sea nets and electrofishing. And those were 2 streams or small rivers, 3 lakes, 3 canals, and 1 ditch system. So to give you an impression, this is a photo of the ditch system. All ditches like this are connected to each other. This is a photo of a lake near Rotterdam. This is a photo of a lake near Amsterdam. This is actually brackish water, so also some marine species occur here. And this is one of the small streams in the eastern part of the Netherlands. So in this study, we also tested our sampling strategy. And schematically, you can see a lake on the left. And in the lake, we took 9 individual samples over 750-meter transects. We sampled 30 liters of water. But then we also, for each 3 individual samples, we took 1 mixed sample with water collected over the 2,250 meters. We also filtered 30 liters out of those. So actually, the mixed samples only have 1/3 of the effort compared to the individual samples. So then when we looked at the results, in this graph, you see the average number of species per sampling strategy that was detected. And when you see on the left, with the 9 individual samples per water body, on average, we had the 18.8 species detected. Then all the way on the right, you see that in the Water Framework Directive monitoring, so the traditional conventional monitoring with electrofishing and sea nets, we detected 20% less species than with the eDNA individual samples. Interestingly, in the middle, you see the results of the 3 eDNA mixed samples. And you see you only catch a little bit less species with the mixed samples compared to individual samples. And we still found detected 18% more species with the 3 eDNA mixed samples compared to the complete Water Framework Directive monitoring, which could be 5 up to even 40 transects. And when we looked at which species were missed in the mixed samples compared to the individual samples, we found that only species that had a very low percentage, low proportion of the eDNA sequences, less than 1% in the individual samples, were missed on some occasions. So in general, all common species were detected with the 3 mixed samples, which is very good. Then we also looked at the effect of the mixed samples on the relative abundance. So what we did is we put the proportion of eDNA per species in the mixed sample against the proportion of the eDNA per species in the individual samples. And we found almost perfect correlations. Only on the top right graph, you see there's one outlier, maybe a piece of fish tissue came into one of the samples. When we take this outlier out, it's also an almost perfect correlation. So to summarize, the 3 eDNA mixed samples give almost the same information as the 9 individual samples, almost the same number of species, but also the same relative abundance of the species. So from there on, we started a really large project in the Netherlands to collect more data with eDNA-metabarcoding to compare to the Water Framework Directive monitoring. And we did that in different water types to see that we cover all different water types. And we also cover the whole gradient of degradation. So the Water Framework Directive gives some quality scores to the water bodies from good to moderate to bad quality. And we are sure to cover all those different qualities in the project. So, and the goal is to use the eDNA metabarcoding data to see if we can calculate indices with it. Within the project, we also seek a collaboration within DNAquanet. We do that by sharing knowledge, data, and we also collaborate on projects. And we do that via the working group 2 that was focusing more on indices, and also working group 3 that was focusing more on methods. So here a brief overview of the project, or this 83 water bodies are sampled in the past 3 years. And you see we divided them into groups. So stagnant linear waters like canals and ditches. We have lakes, we have brackish waters, and we have streams and rivers. So you see it is quite well covered all over the Netherlands. So back to the indices. So in this joint publication of DNAquanet, we made a schedule that gives the key steps of the traditional biological monitoring and assessment procedures. And we show how you can do this with eDNA. So the sampling can be replaced by eDNA samples. The sampling preparation, it can be replaced by the extraction, the PCR, and the next generation sequencing. Then the species identification and quantification, it can be replaced by the bioinformatics. And then you come to the index calculation. And here's the question, can you renovate the indices or can you rebuild them? I'll get back to that later. So first about the indices in the Netherlands for fish. So what is in the current indices? Which data we have to put in? So we put in the species composition, so the total number of species, but also the relative number of indicator species. We can put in the species abundance. This is also the relative biomass of indicator species and also the relative number of indicator species. And finally, in some of the indices, we have age or length distribution. For example, for the pike perch in lakes, we want to see if there's the big females, the big adults, because it's commercially fished. So only in a healthy system, you also find the big ones. And in the estuary, we also do length distribution for the smelt and the twait shad. So what about eDNA metabarcoding? Can we provide the data that is needed? So for the species composition, well, as we've seen in this study, but also in many other studies and articles, the species composition is really good because we have higher detection chances. So this can be done very well. Then species abundance. Well, the species abundance, we get the relative abundance in eDNA sequences, and we can reproduce them very reliable. So we have to see if we can fit them into the indices and how they relate into the relative biomass and relative numbers per species. But I think this can be done too. And then finally, the age length distribution, we cannot reproduce that with eDNA. So then back to the 2 options. Well, option 1 would be to renovate. So that means that we would use the current indices with the eDNA data in it. Well, the advantages are clear. We can reuse the well-validated indices. It will be easy to implement. There's no large changes needed to be done, no new indices needed to be calculated. However, the disadvantages are that those indices are not designed for eDNA data. So we don't make optimal use of the strengths of the eDNA metabarcoding. And another disadvantage is that traditional methods, have their limitations too. So which makes comparing the results of the eDNA metabarcoding more difficult, because we don't know if the traditional methods give exactly the species composition, for example. And in some cases, like shown on the length distribution or the age, it's not possible to generate this data with eDNA. So we have to get some adjustments to the indices. But this could all be done with renovating. Well, if this is not going to work, then the second option would be to rebuild the new indices based on eDNA data. Well, the advantages are, of course, using the strengths of eDNA. For example, the species composition, as we have far higher detection chances, we can include some rare indicator species. Now in the Netherlands, most rare species are not in the indices because they're too difficult to catch, so it's not reliable to put them in the indices. But if we have reliable data on them, we can use them. And we can also account for the limitations of eDNA. And the disadvantages is that it's much more work to calculate the new indices, to find which species would be indicative for the quality scores. And the implementation will also be much more difficult because there's much more work need to be done to make these new indices, and then also to intercalibration with other countries. So in our project, the steps that we follow is we first collected all the data. Now we are going to see if this data can fit in the current indices and if we can produce the same scores. And if not, we will give advice on which parameters in the eDNA could be used possibly to make new indices. So we collected all the data in the past 3 years, and this year we will analyze the data. And at the end of this year, we hope to have the report ready. Are there any questions?